It's now my great pleasure to introduce our next speaker, who is Dr. Sweta Sikder. Sweta studied at the University of Calcutta, where she obtained a bachelor degree in microbiology and a master in biotechnology. She then moved to the transcription and disease laboratory of Tapas Kundu Lab for a PhD. After completing that, she moved to the lab of Yamin Lal at the NIH, where she is a visiting postdoc working on central biology, something I guess we'll hear more in a moment. Sweta, we're very happy to have you here. The floor is yours. I think you are muted.
Yes, can you hear me now?
Yes, that's perfect.
Okay. Good afternoon, everyone, and thank you for this. I thank the organizers for giving me this fantastic opportunity to present my postdoctoral work, which is majorly I have done in Dr. Yamini Dalal's lab, and it involves how the centromeres behave as we age, which you can see here demarcated in red spots. But before I dive into how more about the centromeric proteins, I would like to give a brief introduction of what is aging, and particularly what is cellular senescence, which is a major hallmark of aging. So simplistically, if you take a young individual, which is me here still as a postdoc, so the cells or the nuclei here is actively dividing. But with the erosion of time, there are several progressive decline of several processes like the accumulation of the mitochondrial loss, telomere attrition, accumulation of DNA damage due to the impairment of the DNA repair pathways, and also misregulated autophagic processes, which leads to the exhaustion of the replicative potential of the cells, and they gradually arrest at G1 or G2 phase. And these cells are called the senescent cells. So these are quite unique as they do not undergo apoptosis, but they secrete a lot of cytokines and chemokines, as is shown here, and which further influence the microenvironment of these senescent cells. And what is interesting is that they have a distinct nucleus structure, which I will talk about in my talk more. And the accumulation of this senescent cells leads to organismal aging, as you can see here. This is me, aged, but of course I took help of AI. So when I joined the lab, I was particularly interested in what is happening inside the nucleus of the senescent cells and whether these are the drivers of senescence and thereby organismal aging. So as I know that this audience do not need really an introduction of how the chromatin is organized in the nucleus, but to emphasize that this chromatin organization inside the nucleus changes as one progresses along their lifespan. So if you can see here, in a young nucleus, the chromatin is organized distinctly in specific territories called the lamina-associated domains and the topologically associated domains called the TADs. And there are specific loci which are methylated, and this genetic and epigenetic milieu actually dictates gene expression. But as one moves across the lifespan, there is a reorganization of this chromatin structure. There is the formation of senescent associated LADs, and also demethylation of specific loci, especially of the non-coding RNAs, which leads to the prior methylated or repressed genes, such as the lines and the signs. And gradually, as one becomes old, there is this formation of specific senescent associated heterochromatin foci. So this is prior to the notion of a global loss of heterochromatin. We now understand that there is the formation of specific senescent heterochromatin foci, and they have distinct epigenetic features. So despite there being a plethora of literature or studies about how the chromatin is organized inside the senescent nuclei and whether it dictates the phenomena of senescence or cellular aging, there is a lack of information on how specifically the nucleosomes are organized in the aged individuals. So a prior study from our lab has shown that taking nucleosomes from young and aged livers, where we subjected them to AFM microscopy and showed that there is a difference between the nucleosomal repeat length, which measures the distance between one nucleosome to its neighbor, and it has been shown that the aged liver nuclei has a reduced nucleosomal repeat length. So we were more interested in what was happening at the centromeric foci, because these are foundations for the kinetochore formation and binding of the microtubules, which further leads to the chromosomal segregation. And if you consider the centromeric chromatin, this is quite distinct, because here the histone H3 nucleosomes are replaced by the centromeric histone H3 variant called the CENP. And this further binds to the inner kinetochore proteins called the CEMC, CENB, which directly binds to the centromeric DNA. And the centromeric DNA is a repetitive AT-rich DNA, which is known to get transcribed by RNA pol 2, and which gives rise to this non-coding RNA called the alpha satellite RNA. The CENP nucleosomes are interspersed with the canonical H3, which is modified, especially at K4 dimethylation and K36 dimethylation, which I will speak extensively in my presentation. So studies from our lab and others has shown that this transcription of this non-coding RNA from the centromere is actually essential for the deposition of CENP at the centromeres, which then further forms the basis for the kinetochore formation and thereby binds to the microtubules and leads to the equal segregation of the chromosomes. So what we were curious about, that as cells stop to divide, what is happening at the centromeres? So to answer this, we took help of skin fibroblast obtained from from various individuals, and then we assayed for what is happening to the CEMC expression, which is, as you can see here, is an integral part of the centromere. And as we monitored the expression of CEMC across this vast lifespan, across various lifespan of or ages of the individuals, we see that CEMC significantly reduces as one ages. And I will bring your attention to this two particular age, because this fibroblasts were obtained from the same individual, and this did show that CEMC is significantly downregulated as the individual's age. So taking a cue from their interface nuclei data of CEMC expression, we further wanted to see what is happening at the metaphase chromosomes. So these are metaphase spreads taken from a young individual of 1 year old. And as you can see here, we see the distinct presence of CEMC at the centromeres of the chromosomes. But as one ages, and the 47-year-old individuals, we see that their metaphase chromosomes lose CEMC. And not only this, as you can see here, the chromosomes look more disorganized, bulgy. This also is kind of in lieu with previous observations that aging leads to a global loss of chromatin architecture and loss of heterochromatin. So as it is a technical challenge to work with various skin fibroblasts, as they have a limited lifespan. We chose to see what happens to the expression of the centromeric proteins in vitro senescent models, where we aged skin fibroblasts by passaging them until they reached a high population doubling time, or by treating them with a drug called doxorubicin, very, very low dose, exposing them for 24 hours and then allowing them to grow or senesce for an additional 10 days. So what we see that in both the senescent models, there is a dramatic decrease in CENP, both CENP and CEMC proteins. So to have an overall idea of what is happening to the proteome expression of these cells, we we isolated the nuclear proteins and subjected them to mass spectrometry. And this volcano plot shows the overall changes in the proteome expression in young versus age cells. The red demarcates the proteins which are significantly upregulated, while the green represents the downregulated proteins. And we can see that SEMC was one of the significantly downregulated proteins which we found in the age cells. So to further understand that how these proteins are expressed in different chromatin states, we isolated through salt fractionation the different euchromatin and heterochromatin fractions. And this actually shows the distribution of mostly the histone variants, as well as the centromeric proteins, which you can see here. So what is interesting that other than the significant downregulation of SEMC, what we find is that SEMC which was like originally highly expressed in the heterochromatin fraction in young cells, is now being expressed mostly in the euchromatin fraction. We also find that SEMB, which I will talk later in my presentation, is still present, not like SEMC. It is still present in the age cells, but mostly in the euchromatin fraction. Another protein which we found extensively getting upregulated is p53 as is shown here, and also in this distribution, and also from our RNA-seq data, we found that p53 pathway was significantly upregulated in the age cells. So what we thought is that, does p53 regulate SEMP expression? So to answer this, what we did is we did a knockdown of p53 in aged cells and looked at SEMP expression both at the RNA level by doing an RNA-seq, and we did see that SEMP mRNA levels were upregulated upon p53 knockdown. And this also reflected in the protein levels where SEMP was upregulated. So we were very encouraged by this observation. And we wanted to see that whether this rescue of SEMP expression restores mitosis in aged cells and whether the cells are able to grow back again. So to do this, what we did a double thymidine block and monitored the cells undergoing mitosis. So as you can see here, these are young cells which are undergoing mitosis, and you see the equal chromosomal segregation. There is expression of SEMC and SEMB. But as cells age, we do see a lot of cytokinetic defects, and you see that there is a drastic reduction in the SEMC expression. But when we did a p53 knockdown, we did see that the SEMC expression was rescued. But surprisingly, we still saw that the nuclei in the p53 knockdown cells still had a lot of mitotic defects. So this made us to think that what is actually happening? We are getting back centromeric protein expression, but still the cells are not being rescued of their mitotic defects. So to analyze that, we closely looked at the metaphase chromosomes obtained from young and aged cells. As you can see here, the aged metaphase chromosomes lose SEMC from the centromeres. But when we looked closely at the p53 knockdown metaphase chromosomes, we found that although there is a rescue of the SEMC expression, we found that there is deposition of SEMC or SEMP at the chromosomal, at other chromosomal sites other than the centromere along the chromosomal arms. So this was quite intriguing, as this is a kind of a characteristics which is also observed in cancer cells. So then we wanted to ask that why this is happening. So to understand that, we then looked at what is happening at the centromeric transcription. As I have said earlier, that centromeric transcription or the centromeric transcripts are one of the crucial factors which help in the deposition of SEMP at the centromeres. So what we find here is that indeed, in the aged cells, there is a loss of the centromeric RNA, which is not rescued upon p53 knockdown. So what we understand now is that in the aged cells, there is a loss of centromeric proteins from the centromeres as well as the non-coding RNA transcription, which thereby abrogates the further recruitment of SNP at the centromeres. When we do a p53 knockdown, we do get back the centromeric protein expression, but this, due to the lack or the absence of the centromeric non-coding RNA, gets deposited along the chromosomal arms, and this might lead to the abnormal mitosis. So the next obvious question what we asked is, can we then reactivate the centromeric transcription? So to answer this, we went back to a traditional question, is that how centromeric transcription is regulated. So for that, we again examined the centromeric chromatin. And what we understand is that the centromere, as I have shown you earlier, that the SNP nucleosomes are interspersed with the H3K4 dimethylation nucleosomes. And this H3K4 dimethylation nucleosomes might act as a kind of a transcriptional activator, which drives the RNA transcription. So what we hypothesized is, so what we wanted to see is that how the epigenetic change or epigenetic makeup is changing at the centromeric chromatin as a cell's age. And we do see that there is a stark decrease in the H3K4 dimethylation mark at the centromeric chromatin, and also an increase in the repressive H3K9 trimethylation mark. So what we hypothesized is that maybe there is a lysine demethylase which is getting recruited in the aged centromeric chromatin, which diminishes the H3K4 dimethylation mark. So if we now have an inhibitor of the lysine demethylase, can we rescue the centromeric transcription? So for that, we took help of the small molecule inhibitors of lysine demethylase, and we subjected both the young cells and the aged cells to this lysine demethylase inhibitor. And what we find is that the lysine demethylase inhibitor had no such effect on the centromeric transcription in the young cells, but in the aged cells, it led to a rescue of the centromeric transcription. And this was further validated upon p53 knockdown in the aged cells, where we found that upon lysine demethylase inhibition, the centromeric transcription was rescued. So we were very excited, and we wanted to see that what is happening now to the mitotic defects which we observed in the aged p53 knockdown cells. So as you can see here, these are the p53 knockdown cells which showed a lot of mitotic defects, majorly the cytokinetic defects, or the chromosomal lagging. But when we treated them with the lysine demethylase inhibitor, we found that there is quite a rescue of the mitotic defects. As you can see now, equal segregation of chromosomes and no more presence of the cytoplasmic breaches, as you can see here. So now we successfully have established a system in the aged p53 knockdown cells where we both have centromeric protein expression as well as a centromeric RNA transcription going on. So the last question what we asked is that what is actually recruiting the LSD1 at the centromeric chromatin? So as I have showed you earlier, despite the loss of CEM-C and CEM-P, the CEM-B protein, which directly binds to the centromeric chromatin, is still present at the centromeric, is still present in the aged centromere. So we ask the question, does this have anything to do with the recruitment of the lysine demethylase at the centromeric chromatin? So first, what we did is we checked whether the expression of CEM-B in young versus aged cells. And as you can see that despite the downregulation of CEM-P, we see that the CEM-B levels do not change. We also then adopted the knockdown method and knocked down CEM-B in the aged cells. And here we did see that there is a rescue of the centromeric transcription, as was seen in the lysine demethylase inhibition. To show or to validate whether this rescue of centromeric transcription is happening through the H3K4 dimethylation pathway, we further checked the enrichment of H3K4 dimethylation through ChIP-qPCR at the centromeric chromatin. And as you can see here, that we indeed see that when we knock down SEN-B, there is an increase of the H3K4 dimethylation transcriptionally active mark, which leads to the centromeric upregulation of the centromeric RNA. And this is quite specific for at present or happens at the centromeric DNA. So this brings me to the summary of my talk. So what we understand here is that in young cells, we have an intact centromeric chromatin where we have SEM-PA nucleosomes, SEM-C proteins present, SEM-B binding to the centromeric DNA. There is H3K4 dimethylation nucleosomes interspersed with the SEM-PA nucleosomes, and this leads to the active transcription of centromeric RNA, which in turn helps in the SEM-PA recruitment at the centromeres and helping in the binding of the microtubules and thereby leading to accurate mitosis. But as cells age, there is an upregulation of p53, which leads to the depletion of SEM-C and SEM-PA proteins. And there is also the recruitment of lysine demethylase, which abrogates the H3K4 dimethylation mark, thereby leading to a downregulation of centromeric RNA. When we do and this leads to the abnormal mitosis. When we do a knockdown of p53, we do rescue back the centromeric protein A expression, but it is still not getting recruited at the centromeric chromatin because of the low centromeric RNA. So when we inhibit the lysine demethylase, we again have we bring back the H3K4 dimethylation back at the centromeric chromatin, thereby leading to the upregulation of the alpha-SAT RNA. And now the centromeric SEM-PA nucleosomes are back at the centromeric chromatin, building an intact centromere, a functional centromere, which then can lead to an accurate mitosis. So with that, I end my talk, and you can check out my preprint and bioarchive. But before I end, I would like to show my most important slide. This work was done in Dr. Yamini Dalal's lab, and I thank my mentor for her support and critical inputs in this project. I have a fantastic supportive lab colleagues here who has supported me throughout since I joined the lab, especially during the COVID times. I also had a fantastic summer project, summer student join me, Truman, who have been helping me with the work I showed here today. I thank Songjun for all the bioinformatic analysis and all the core facilities at NIH and CI, and also Talos and Alex for the mass spectrometric analysis. Thank you, and I will now take questions if there is any.
Thank you, Sweta. Fantastic talk. Very, very clear and linear. And you have a couple more general question if you wish just to contextualize a bit more the talk for a bit better understanding. So is there any and if yes, which one interaction of this pathway that you described with the other hallmarks of senescence that you described in the beginning? And also if there is any parallel or clear difference that can be drawn with other cell types that do not divide or divide very slowly, but are not in senescence, like neurons or, this kind of stuff or cell types.
Yeah, so I will answer your second question first about the cell types. We have not checked actually in stem cells or neuronal stem cells particularly, but I did check in like different kind of fibroblasts and also cells from epithelial origin. And I have seen that centromere proteins decline as these cells age. For your first question of understanding other senescence markers, if I got the question correct. So I did not show this the data due to time constraints that I knocked down SEM-PA particularly in young cells. And I see that within 72 hours, there is an onset of senescence, that lamin B1 degradation, high beta-gal present in the nuclei, and the cells actually do not recover, and even if you remove the ASOs, which is targeting SENPE, even after it takes almost like 2 weeks for the cells to again recover back. So targeting only SENPE also give this kind of senescent markers, upregulation of the senescent markers.
And instead, what have you tried to have a sustained SNP expression and ribosome centromere transcription earlier on as a preventative measure to senescence?
Sorry, can you repeat it?
What I mean is that have you tried to, instead of rescuing the senescence phenotype, to prevent it by maintaining expression of the centromere proteins and the centromere RNA? Do
Yeah,
the cells still age, or is the senescence process slow down?
yes. Also that data I did not show that, but those experiments were done only after inducing the senescence, where we did a p53 knockdown, and we did see that we did the S phase staining and saw that the cells again replicated back. But to answer your question, whether to maintain the SENPE levels in the young cells, whether if we keep on maintaining this SENPE or the centromeric transcription levels in the young cells, I have not done those experiments, but those are quite interesting ones, which I can look into.
Thank you very much.
See a hand raised.
There was a question in the Q&A, which
Yes.
I think you mostly answered already, but was what happens to the ectopic deposition of SENPE and SENPC during the p53 silencing and reactivation of centromeric transcription? So mostly focusing on the ectopic deposition.
So we have now initial data suggesting that when we rescue back the centromeric transcription, we are actually bringing back, or I mapped the actual CEN-B and CEN-P co-immunofluorescence, and they are coming back. So, but yes, I am planning to do a ChIP-seq. I have done the ChIP-seq and in the young and the age cells, but I want to further map the exact chromosomal locations of the SENPE protein and see whether it is completely rescued back upon the centromeric transcription reactivation. Thanks.
Thanks. And we just had another question because
Yes.
thanks for the great talk. Did you see any H3K36 methylation level change at the centromere during the
Yes, thank you for the question. I actually checked a couple of epigenetic marks, not specifically H3K36. I checked H3K9, K27. So yes, maybe I should, yes, that's a good suggestion. Maybe I should also look into H3K36 as they are also present at the centromeres. I see a hand raised by Anirban.
Oh yeah, good catch. Anirban, I will allow you to talk. You should be able to ask your question now. Anirban, I think you can.
Yeah, can you hear me?
Yes, now yes.
Okay, so great talk. I was just curious to ask you, did you find any particular factors localizing in the chromatin bridges that you showed us in the artificially depleted condition, apart from the centromeric histone variants?
Oh, okay. That's a great question. I actually have not looked into when I did the staining. I mostly looked into CEN-B and CEN-C. I did find CEN-B staining there, but not the other factors. But I also kind of looked at H3K9 trimethylation, but I don't see deposition at the chromosomal breaches, as you said.
Right. I have one more question.
Yes.
Does LSD1 have any function in regulation of transcriptional termination in the p53 depletion context?
So LSD1 has been implicated mostly in transcriptional repression by usually removing the H3K4 trimethylation or dimethylation mark, but particularly in termination, I have not seen any literature on that.
Oh,
But
okay.
that's a great one.
Thank you.
Yeah.
Okay, fantastic. Thank you very much for the talk again and
Thank you.
great answers. And I